Okay, well, I'll get going on my presentation. I'd like to talk to you about immune regulation in the intestine. And of course, we've been considering over the last, first of all, I should thank the organisers very much for inviting me along. It's been wonderful meeting and hearing these different perspectives, people coming from different angles. And of course, that is the challenge for the field, to be able to integrate this different information as we move forward. So I'd like to talk about immune regulation. And of course, we've been considering the fact that normally most of us reside peacefully with our abundant intestinal flora. But of course, as in any relationship, things can break down. And in inflammatory bowel disease, there seems to be disruption of this mutualistic relationship with the development of chronic intestinal inflammation. And of course, research in inflammatory bowel disease has had a very exciting decade. There's the results coming in from genome-wide association studies, clinical studies, as well as information from mouse models. And I think there is some convergence of this information, suggesting a multi-step, multi-hit type of model for the development of inflammatory bowel disease, where there are alterations in the handling, the barrier function of the intestinal epithelium, and how the host is able to contain its abundant intestinal microbiota. And of course, pathways involved in that have been identified in the genome-wide association studies, including those such as NOD2 and genes controlling autophagy. But there is also evidence for alterations in the adaptive immune response that may be linked to changes in innate immunity, with changes in the balance between effector and regulatory type populations. And then, of course, there are alterations in the intestinal flora, and it's difficult really to understand what is primary and what is secondary when one tends to be looking at patients when they already have established disease. But it seems quite clear that there's just not one predisposing factor. You need environmental triggers taken together with alterations in both innate and adaptive immunity. And I want to focus particularly on this balance of the effector and regulatory cells in terms of intestinal homeostasis. As there are many microbiologists in the audience, I thought by way of introduction, many people have spoken about regulatory T cells. Just to remind you, the FOXP3 variety is a specialized population of T cells that are selected in the thymus. They acquire the transcription factor FOXP3 from the fork-head winged helix factor family, and this plays an important role in conferring their function, which is to be able to suppress maladaptive effector-type responses. Not only are there thymic-derived regulatory T cells, but peripheral T cells under the influence of TGF-beta can acquire a regulatory T cell phenotype. And of course, the relative contribution of the so-called natural versus induced is the focus of a lot of investigation. There's been abundant evidence from our own work, the Sakaguchi lab, Rudensky, and many others that regulatory T cells play an important role in immune homeostasis. But I think this is revealed particularly by the phenotype of IPEX patients who have a loss of function mutation in FOXP3 and suffer a potentially fatal inflammatory disease, manifesting itself as type 1 diabetes, allergy, as well as intestinal inflammation. So evidence that the regulatory T cell pathway and FOXP3 plays an important role in immune homeostasis in humans. It's of interest then to understand the factors that control the balance between regulatory and effector type populations, because if we want to be able to manipulate the immune response, this is important information. We've been focusing on this in the context of the intestine, and I think there are going to be tissue-specific elements that control regulatory function and activity. We've been particularly interested in myeloid cell populations in that context. It's also clear that the intestine is a preferential site for the induction of FOXP3 positive cells from naive precursors under the control of specialized dendritic cells, retinoic acid, and TGF-beta. So how this process is controlled and what promotes inflammation versus control of this is important to be able to to dissect. So I'd like to, in the context of this, then discuss the role of IL-23, which has been introduced very nicely by Gérard Ibel in controlling both innate and adaptive effector functions in the intestine, and finish with a few comments about the regulatory T-cell axis. Now, IL-23 is a member of the IL-12 family of cytokines, brought to our attention, I think, by some of the pioneering work by Dan Quah and Rob Castelline at DNAX. Now, like interleukin-12, IL-23 is produced by activated myeloid cells. There's a skew towards IL-23 production by myeloid cells in the intestine relative to other systemic sites. The IL-23 receptor, a component of which is shared with the IL-12 receptor, is expressed on T cells as well as innate immune cells. And we tend to think of IL-23 as being important in promoting Th17 responses. And again, these were introduced to you over the course of the meeting. And I'd just like to make the point that, of course, Th17 cells are very abundant at mucosal surfaces and play a very important role in intestinal barrier function and antimicrobial activity in the intestine, I think under both homeostatic and inflammatory conditions. And of course, some of the key cytokines that are produced by Th17 cells are particularly tailored to this type of antimicrobial defense. So interleukin-22 will act on non-hematopoietic cells such as epithelial cells through activating STAT3, leading to epithelial cell restitution and the production of antimicrobial peptides. Interleukin-17 is able, again, to act on non-hematopoietic cells, leading to the production of a number of chemokines that will promote the accumulation and recruitment of neutrophils into the inflammatory site. So important for functions in terms of host defense. But you can imagine if this pathway is triggered in response to a microbe that we're not able to eliminate, then things can become pathologic. So this pathway of IL-23-driven immunity provides important host defense, but can flip over into inducing immune pathology in certain settings. And being able to understand what controls that may give us some clues in how we could more specifically target this response. And indeed, there's very good evidence that IL-23 is involved in a spectrum of tissue inflammatory diseases. It has been shown in a number of model systems, including colitis models that I'll show you, but also joint inflammation, CNS inflammation, and skin inflammation, that IL-23 plays a pathological role in driving this response. In inflammatory bowel disease, there's evidence in some patients for increases in Th17-associated cytokines. And what has also emerged from the genome-wide association studies is the identification of multiple SNPs in the IL-23 receptor pathway that confer risk to inflammatory bowel disease in both Crohn's disease and ulcerative colitis. And this includes the IL-12p40 component of IL-23, the IL-23 receptor, downstream signaling molecules such as JAK2 and STAT3, a chemokine receptor associated with raw gamma T expression, CCR6, as well as a number of the Th17 signature cytokines, such as IL-17A and IL-17F. Now, how is IL-23 working to promote Th17 responses? I think that has not been very well understood. There's evidence from Dan Quah's work that IL-23 promotes effector function amongst the Th17 population. I'd just like to draw your attention here to a sort of a dichotomy between the Treg cells that I mentioned to you that are FOXP3 positive and these Th17 cells, in that both of these populations have a dependency on TGF-beta for their development. So they're related in that way, but depending on the presence of STAT3 signaling cytokines, that can favour the development of Th17 versus the FOXP3 populations. And this has been studied particularly in vitro, but precisely how this is controlled in situ in different inflammatory lesions is not very well understood. So over some years now, we have been interested in elaborating the role of IL-23 in intestinal inflammation, and our first evidence in collaboration with Dan Qua some years ago was to ask the question whether it was IL-23 or IL-12, the related cytokine that was required for the development of chronic intestinal inflammation in the T-cell transfer model. So transfer of naive T-cells in the absence of a regulatory T-cell compartment leads to a dysregulated response to the flora in immune-deficient mice. And this disease has the hallmarks of both Th1 and Th17 type responses and is dependent on IL-12p40, interferon gamma, and TNF-alpha. So we asked whether that was the IL-12p40 was a component of IL-23 or IL-12, and we're able to show that IL-23 was crucially dependent for the development of chronic intestinal inflammation in this model. When we transferred calitogenic T cells into immune-deficient mice specifically lacking IL-23, they did not develop colitis, whereas if they were transferred into mice specifically lacking IL-12, then they did continue to develop colitis. But what was clear was that IL-23 was not required for the development of the systemic inflammatory response. So this was a compartmentalization of this immune pathological response, with IL-23 promoting mucosal inflammation and IL-12 being sufficient to drive the systemic signs of this disease. Now, associated with the lack of colitis or impaired colitis in the absence of IL-23 was an increase in FOXP3 positive cells in the intestine that we showed played a functional role in suppressing disease. So one pathway through which IL-23 promotes intestinal inflammation is by preventing the development of FOXP3 positive cells locally in the intestine that would inhibit the disease. So how is IL-23 working in this setting? The IL-23 receptor, as I mentioned, is expressed on T cells as well as innate leukocytes. So to address that, we asked the question whether IL-23 receptor deficient T cells would be capable of inducing disease in this model, and the answer to that was that they were markedly reduced in their capacity, one, to accumulate in the intestine, and two, to actually give the signs of colitis. So in the absence of IL-23 receptor signaling into the T cells, there's a marked reduction in the ability of these cells to be able to accumulate in the intestine, and that leads to a reduction in the number of both Th1 and Th17 cells. But I would point out that this effect, again, is highly specific. It's not that IL-23 receptor deficient T cells cannot repopulate the immune-deficient host. They expand extensively and cause the splenomegaly and wasting disease. You need IL-23 to actually promote this immune pathological response in the intestine, something beyond what IL-12 is capable of doing. So how is IL-23 influencing immune cell effector function? I've told you that it impedes the accumulation of calitogenic T cells in the intestine. So to address this, we've looked at the differentiation of the cells that do get to the intestine following transfer of IL-23 receptor deficient T cells. And actually what was quite striking is that there's really no change in the frequency of cells capable of producing interferon gamma or interleukin-17 compared to IL-23 receptor competent T cells. But one thing that was different was that this population of cells expressing both interleukin-17 and interferon gamma was markedly reduced in the absence of IL-23 receptor signaling into the T cells. In addition, there was a reduction in the capacity of all these effector populations to be able to proliferate locally in the intestine, as assessed by expression of the proliferation antigen Ki63. And by contrast with the reduction in this effector population, there was an increase in the frequency of FOXP3 positive cells in the intestine. So we think then that IL-23 receptor signaling into T cells plays an important role in shaping the immune response in the intestine, in promoting the accumulation of T cells and in influencing their effector function. And we did some experiments that would allow us to assess the cell intrinsic or extrinsic role of IL-23 receptor, and I'm not showing you those, but those results suggested that the capacity of the IL-23 receptor to promote this emergence of a sort of hybrid Th17, Th1 population was a cell intrinsic activity, as was the ability of the IL-23 receptor to reduce the frequency of FOXP3 positive cells. So IL-23 receptor shaping the nature of the effector T cell response. And of course, this population of double positive IL-17 interferon gamma secreting T cells has been the subject of study in a number of models of immune pathology, and there's also evidence from the work of Annunziato and colleagues, as well as René Duval at DNAX, that in inflammatory bowel disease there is an increased frequency of these double positive populations of cells. And in addition, of course, there's a suppression of the FOXP3 development. So this is talking about how IL-23 receptor signals into T cells and influences an immune pathogenic pathway in response to intestinal bacteria. We also have over the years developed a number of different models of chronic intestinal inflammation, and one of these is the Helicobacter hepaticus model that develops in genetically susceptible 129 rag mice following infection with Helicobacter hepaticus, a natural mouse pathogen. Now, in this setting, of course, there are no T cells in the mice, but what happens is these mice develop a chronic intestinal inflammation, a prominent cecal infiltrate, which can, on the susceptible background, progress to colon cancer. And we were able to show in early studies that this was inhibited by an antibody to interleukin-23, and it is also inhibited by a number of the cytokines that have a role in driving the T-cell dependent disease, including interleukin-17A and interferon gamma. And in fact, if you look in these rag mice that are infected with Helicobacter in the intestines of the infected versus uninfected controls, then you see a marked increase in these Th17, Th1 associated cytokines in these RAG deficient mice. So, and this was IL-23 dependent. So this alerted us to an innate pathway through which IL-23 induces immune pathology. And Sophia Bonacore in the lab spent a number of years trying to be able to dissect what these IL-23 responsive population was, and to cut a rather long story short, she was able to show by sorting various immune cell populations from Helicobacter hepaticus infected mice and stimulating these with interleukin-23, this would lead to a detectable production of interleukin-17 as well as interferon gamma, and she was able to show that the responsive population resided within a Lin-negative population, but expressed a number of markers that have been associated with the innate lymphoid cell populations that Gerard Eberl mentioned to you. But there are some distinct differences in this ILC population in that it is c-KIT negative and CD4 negative, as well as negative for various NK markers. But it does share the features being IL-7 receptor alpha positive, CD25 positive, and raw gamma T positive. And this population, when isolated from the rest of the leukocytes, was markedly enriched in its capacity to produce a variety of Th17, Th1 associated cytokines. So this population, which we described phenotypically as being Thy1 high, SCAR1 high, accumulates in the inflamed intestine of immune-deficient mice with Helicobacter-induced colitis. So the question was whether it was playing an immune pathological role in the disease. So Sophia administered neutralizing, depleting anti-Thy1 antibodies and was able to show that that is sufficient, that depletion of this population was sufficient to inhibit the development of disease and reduce these inflammatory cytokines. So then from this model, we have identified an innate cell that's involved in the tissue inflammatory response. It responds to interleukin-23 with the production of IL-17 and interferon gamma. In other studies, in another model, we were able to show that this activity is raw gamma T dependent, and therefore this population resides in this expanding family of innate lymphoid cells. I think they may play a very important role in shaping immunity in the intestine. They are poised in the lamina propria, ready to be able to respond to microbially induced signals, be they epithelial-derived or, in this setting, derived from myeloid cells, and they therefore may contribute both to organogenesis and inflammatory cytokine production. So once there is IL-23 being produced in response to microbes, this will stimulate these innate lymphoid populations, which in the absence of appropriate control may also acquire this immune pathogenic phenotype with interferon gamma secretion, IL-17, promoting a host of inflammatory innate cells that can lead to a cycle of positive feedback and chronic inflammatory disease. This is a slide given to me by Jürgen Spitz. Of course, there have been a lot of interest in these populations of innate lymphoid cells. I won't go through all of these because Gerard has outlined them to you, and Jim Santo will talk about this in more detail. But the point I'd like to make to you is that there seems to be immense plasticity in these populations that is adapting to ongoing conditions in the intestine, and they may play an important role not only in host defense but also in precipitating the flip over into chronic inflammation. And of course they're a variant of the classic lymph node inducer type populations, beautifully identified by Rainer Mabius and of course followed up by Gerard Ibel. So this is of course a rag mouse. It's a highly manipulated model. We have to do that to be able to dissect pathways. Is there any relevance of this in terms of chronic intestinal inflammation in humans? So we've wanted to analyse this. Do we find these types of populations in the human intestine, and can they respond to interleukin-23? So Alexander Jeremiah and Carolina Arancibia in the lab have used the same sets of markers and have been able to identify CD127 positive raw gamma T expressing populations in the intestine, and of course Jürgen Spitz and others have shown these types of populations in the human tonsil, and also Marco Colonna identified the NK type expressing ILC that is present in the human intestine. So what Alessandra was able to do was to isolate CD3 negative populations from the intestine or non-T, non-B cells, and stimulate these with IL-23, and can show a marked increase whether we are isolating these from either control or inflammatory bowel disease patients in IL-22. This is produced by both the control and the inflammatory bowel disease patients, whereas the capacity of IL-23 to promote IL-17 was something that was primarily contained within innate lymphoid cells from the inflamed intestines of IBD patients. And if you actually quantitate different subsets of innate lymphoid populations, then Crohn's disease specifically was associated with an increase in the NK negative LTI-like populations, which we did not see in ulcerative colitis. So evidence that IL-23 responsive innate lymphoid cells are present in the human intestine, and there may be changes associated with different forms of inflammatory bowel disease. What I want to draw your attention to in this slide then is really a remarkable conserved axis of IL-23 raw gamma T function in both innate and adaptive lymphoid cells in the mouse and the human intestine. I think under homeostatic conditions, there's an important role for these populations of cells in promoting host defense and barrier function, but upon sustained bacterial-driven responses, IL-23 can promote the elaboration of a more pathological type response, leading to the production of not only IL-17 and IL-22, but also interferon gamma by these populations of cells. And there's some evidence that there's a selective increase in these populations in Crohn's disease. And I think understanding how IL-23 may influence this pathogenic program, what cofactors are involved, what bacterial triggers promote this, may provide some more specific targets, because of course any target that takes this axis out completely may be deleterious in terms of inhibiting chronic intestinal inflammation. So just to finish with a couple of points about regulatory T cells, I've told you about IL-23-driven colitis, and transfer of regulatory T cells is able to prevent the development of this disease, and we've identified a variety of the mediators that are involved here. And I would just make the point that regulatory T cells can control this T cell independent innate lymphoid cell activity. But I suppose importantly from a therapeutic standpoint, can regulatory T cells manipulate an ongoing intestinal inflammation? Chris Motet did this experiment when he was in the lab and was able to show that transfer of an inoculum of regulatory T cells into mice with established colitis was able to ultimately bring the inflammation into control and ameliorate and restore normal intestinal architecture. And the ability of regulatory T cells to be able to do this in this model is highly dependent on their capacity to produce interleukin-10. And here I just want to make a point to you that there are regulatory T cells and regulatory T cells, and there seems to be some functional adaptation of regulatory T cells function in the intestine, and that involves their ability to start to produce interleukin-10. So this is looking at IL-10 staining amongst FOXP3 positive cells. If we take these from the spleen, around 5 to 10% of splenic CD4 positive cells are FOXP3, but the majority of them are not capable of producing interleukin-10 upon polyclonal stimulation, whereas there's a larger frequency of FOXP3 positive cells in the colon, and a larger proportion of those are capable of making interleukin-10. So why is IL-10 particularly important? Well, we think that's important in the context of what the mouse models have shown us. But there is now evidence from human populations, families where there is a genetically encoded early onset colitis, very severe early onset enterocolitis. Some of those have been shown to be attributable to mutations in the loss of function mutations in the IL-10 receptor alpha or IL-10 receptor beta genes. So clear evidence in humans that IL-10 is a key player in intestinal homeostasis. So I just want to finish now by just making a point here about Helicobacter hepaticus. I mentioned to you this is a model that we use to elicit colitis in immune-deficient mice. But I think this makes the point of how bacteria can have different functions depending in what setting the immune system is being exposed to them. So Helicobacter hepaticus is a microaerophilic pathogen of mice. It colonizes the intestine in the gel mucus layer. It actually lives in the crypts in very close proximity to epithelial cells. But in normal hosts, it does not elicit any inflammation. But it does prime the immune system because it elicits a systemic IgG response. So it's not a completely innocuous commensal. It could be considered, as Sarkis Masmanian has suggested, as a pathosymbiont. It's endemic in many animal houses, but causes no immune pathology in normal mice, but will induce immune pathology in mice with immune deficiency, and this may be what's happening in IBD patients exposed to certain types of intestinal bacteria. So how does Helicobacter hepaticus prevent the host from overreacting to it? Because it establishes a lifelong colonization. And one of the ways in which it does this is to promote IL-10 production by CD4 positive T cells in the intestine. And in fact, if you neutralize IL-10 at the time of Helicobacter hepaticus infection, then you end up with chronic intestinal inflammation. And this follows on from work originally pioneered by Marika Kohlberg. So regulatory T cells, I think, play an important role in intestinal homeostasis, in part through their ability to produce interleukin-10. The gut microenvironment favours the regulatory T cell axis. There's specialised antigen-presenting cells that promote regulatory T cell induction. Intestinal bacteria themselves seem to promote interleukin-10 production, and there was a recent example of that from the studies of Kenda Honda, but there are others. And Helicobacter hepaticus also induces IL-10 that may be an important way in which form of bacteria-host mutualism and important in the relationship between the host and intestinal bacteria. So is this type of relationship altered in inflammatory bowel disease? And of course, SNPs in IL-10 are evident in both ulcerative colitis and Crohn's disease. So I finish then by reviewing some of the things that we've discussed at this meeting. Clearly, there are lots of different bacteria. Some are more immune stimulatory and others. Some can influence different aspects of immunity. And of course, it's going to be a fundamental challenge to be able to work this out. And of course, is the dysbiosis that is observed in inflammatory bowel disease, where there may be reductions in certain types of bacteria that perhaps are very good at promoting the IL-10 axis. Could we try to manipulate this to help restore intestinal homeostasis in IBD patients? But of course, the challenge with IBD is that it's Crohn's disease and ulcerative colitis, and there's great heterogeneity amongst the patients. So we need to understand what are common pathways and what are more individual type pathways. And we might want to consider different types of treatment depending on where the lesions are. Manipulating the flora, fortifying barrier function, bolstering innate immunity, autophagy and bacterial regulation, and perhaps manipulating these adaptive responses that I've mentioned to you. And of course, patient stratification will be crucial in being able to try to address these issues. I want to stop now. You'll be pleased to hear and acknowledge my collaborators in this work. This has been a long-standing collaboration on IL-23 with Kevin Malloy, Sophia Bonacore, Philip, Anna, Chris contributed to the IL-23 studies. Michael and Julia and Andy Johnson are working on the IL-10 axis, and Alessandra Jeremiah and Carolina Arancibia are taking us into the very difficult area of human immunology. Here we're indebted to our gastroenterology colleagues Simon Travis and Satish Keshav. We've had some wonderful collaborations with individuals who've helped us with reagents, Herman Waldman with the anti-Thi1, the Celtec group for the anti-IL-17, Dan Kwa for IL-23 reagents, and Dan Lippman and colleagues for the raw gamma T deficient mice. Thank you very much.